In this video, I'm going to focus in on the molecule of DNA and how that molecule is replicated, or how DNA replication occurs within the cell. And you can see here is a very lightweight model of what DNA replication looks like. And by the end of these slides, you'll be able to really understand what you're looking at here. So before we go into too much detail, let's really remind ourselves of the structure of DNA. And I've drawn the structure of DNA here, though you've seen this structure before. And you may recall that the molecules of DNA are made up of single units called nucleotides. And so this is a nucleotide drawn here, and a nucleotide is actually made up of 3 units as well. And those 3 units that make up a nucleotide are phosphates, phosphate here in black, a ribose sugar, shown here in green, and then a nitrogenous base, which I've shown here in red just with the terms base, because the nitrogenous base can actually change, and we'll talk about that in a minute, how this base can change. So a DNA molecule, or an RNA molecule for that matter, is made up of a phosphate, a ribose sugar, and a base. And so these go together, phosphate, ribose sugar, base, and they link to one another by the next phosphate to the next ribose to the next nitrogenous base. Now this bonding occurs from one end of the ribose to the other, and these sugars on the ribose are numbered, 5 here, 3 here. So they number going around 1, 2, 3, 4, 5. So the phosphate is going to bind closest to the 5' carbon. The other phosphate is going to bind closest to the 3' carbon. Therefore, the molecule of DNA actually has a direction. It has a direction it goes, and by convention, that direction is 5' to 3'. So when we draw out a molecule of DNA, we're always going to draw it 5' to 3', either top to bottom or left to right. Now, you might remember from earlier when we talked about DNA, that DNA is actually made up of 2 strands of the molecule. There are 2 strands of DNA that make up a full DNA molecule, and these 2 strands are said to be anti-parallel. And what we mean by anti-parallel is that one molecule going from 5' to 3' top to bottom will be paired up with another molecule going from 5' to 3' bottom to top. So these molecules are not the same, the left and the right, but they're flipped over on top of each other. So again, you go 5' to 3', which is the way I've drawn it here. And on the other side, it'll be 3' to 5' top to bottom. So imagine this molecule flipped over 180 degrees and then bound back in to this. This will make more sense in the next slide when I talk about the bases. So as I showed you that molecule of the phosphate and then the ribose and then the base, I left that base area just as a blank area that said base. And what you may know is that this base can actually be filled by 4 different molecules. And these molecules are known as adenine, guanine, thymine, and cytosine. And each one of these molecules binds onto that ribose sugar via this area that I've labeled with the blue arrow in each drawing. Now these structures are complex for each of these molecules. I don't expect you to know these structures, but I just wanted you to see them. To understand that we call these nitrogenous bases because they're full of nitrogens. There's all sorts of nitrogens in here. And these molecules are actually remarkably similar to one another. There are only small differences. We can classify them into 2 major groups. One is called the purines, and the other is called the pyrimidines. And so within the purines, we have adenine and guanine. And you can see the only big difference between these 2 is that you've swapped out an oxygen for a nitrogen here. And you've added a nitrite group here when you don't have one there. Various changes in the molecule. But overall, they look very similar. They have 2 rings. They have things coming off of them. So that's adenine and guanine. You can compare that to the pyrimidines, which have just 1 ring, thymine and cytosine. And again, you can see that overall the structure is the same, but the molecules that are coming off of them are different. Now, I put these in different colors for a very specific reason, and you might have already caught up on this. Adenines always bind with thymines. So a purine always binds a pyrimidine to make that single strand of DNA into a double strand. Adenines always go with thymines. Guanines always go with cytosines. So A and T bind one another, and G and C bind one another. So despite all of these structures, what you really need to remember out of this is that an A and a T will always stick to one another, and a G and a C will always stick to one another. These molecules bind very strongly by hydrogen bonds. So hydrogen bonds are what hold the entire DNA molecule together. These nitrogenous bases and these hydrogen bonds are important both for the formation of the DNA molecule and for the replication of the DNA molecule itself. Now, when you want to replicate a DNA molecule, the advantage of having these base pair rules, having it a rule that A always binds to T and G always binds to C, is that you can use each strand of DNA as a template to make a new strand. And this is known as semi-conservative replication. And so you can see here in the diagram in blue, we have one double strand of DNA, 5' to 3' on the top by convention. The anti-parallel strand is 3' to 5' below it by convention. Now, as we go through replication of this DNA, we're going to add new DNA here in red, and you'll see that the 5', 3' strand stays while a new red strand forms with it. And then the 3', 5' strand stays while a red strand is formed above it. So one molecule of DNA becomes two molecules of DNA. Each of the two resulting molecules have 1/2 of the original genetic material. And just as a reminder here, A always goes with T, and G always goes with C. And that is what really drives the formation of these new DNA molecules, and that's what causes this semi-conservative replication to work. Now, just as a little historical perspective on determining that this semi-conservative replication was in fact the way it goes, I want you to realize that it wasn't always a given that semi-conservative replication was the way to do it. Originally, a lot of scientists thought that one strand of DNA was used as a model for another strand of DNA, but then the original blue-blue strand remained while a new red-red strand was formed, and similar models to that. And it wasn't until Michelson and Stahl performed this experiment, which people tend to call the Michelson-Stahl experiment, and some have dubbed the most beautiful experiment in biology, that really showed, it really demonstrated that semi-conservative replication was the way that DNA replicates. And because this semi-conservative replication of DNA is so critical to understanding how DNA replication occurs, and because this is sort of a foundational experiment in the field, I just wanted to take a moment to explain it to you. So what Michelson and Stahl did was that they grew E. coli. And you'll remember that those bases had a lot of N's, a lot of nitrogens in them. So they grew some of the E. coli in the presence of nitrogen 15, which is a slightly heavier isotope of nitrogen, than N14, nitrogen 14, which is the normal isotope of nitrogen that you can see, that you tend to see in the environment. So by doing this, what they ended up making was one set of E. coli that had DNA that was slightly heavier by virtue of having N15s all throughout it, and another set of E. coli that had DNA that was slightly lighter by virtue of having just N14s in it. Now you might ask yourself, well, why does that matter? Why does it matter that they made one set of DNA heavier and one lighter? Well, Michelson and Stahl also figured out a way to separate these DNA by their weight. And they used a tool known as an ultracentrifuge, and they were able to, in a tube, separate these strands of DNA so that they could see a big mass of DNA. So when they did this with the heavy treated cells, they got a nice band of DNA low in the ultracentrifuge tube. When they did it with the lighter one, they got a nice band higher in the tube. Okay, so are you following along with that for now? So then what they did was they took the heavy DNA, the experiment with the heavy DNA, they washed all of the heavy materials away from those cells, put them back into normal conditions, and then allowed them to start dividing. So there are no more heavy nitrogen except for that which is already incorporated into DNA in these cells. So when they put them back into normal material, after one round of duplication, after these cells were allowed to duplicate once, they no longer saw a band that was heavy down here. Instead, what they saw was a set of DNA that was sort of in the middle. This DNA was in the middle because it had some heavy and some light DNA incorporated. They also saw a band of light DNA, and the reason they saw this was it's pretty impossible, as you'll remember from studying the cell cycle, to stop these cells from going through only one replication. So what they're seeing is that these cells over a period of time have replicated either once or more. If they've replicated more, they've started to see DNA that is completely light. It has no heavy molecules. And as they continue on through time, they keep seeing more and more of this normal weight DNA appearing. And as more and more of this appears, the amount of heavy or mixed heavy DNA that they see is decreasing because it becomes a smaller proportion of the material. Now this experiment is complex, and I can understand if you don't completely understand this experiment, but take a moment to think about it and realize what they showed. They really showed in an experimental fashion that these two strands of DNA now became middle weight DNA when they incorporated these red strands. And then when these divided again, you would now have two sets that were blue and red and two sets that were red and red. And those red and red strands would be light, not middle weight. And so that's what they're seeing over time. So they very clearly demonstrated that DNA replicates in a semi-conservative fashion. So aside from demonstrating this semi-conservative fashion, there's been enormous amounts of work done on how DNA replications occur. And what I want to do is give you the nuts and bolts, the highlights of what's happening when DNA replications occur. And this, I will go ahead and tell you, is leaving out an immense amount of detail. And I'm just trying to give you some of the big ideas that are happening here. So DNA replication occurs during the S phase of the cell cycle. That's the phase where the genome is being duplicated in preparation of dividing the cell. So all of this process is occurring in S phase. And let me orient you a little bit with this drawing. So we have a strand of DNA, a double helix, and I've labeled 5', which goes all the way up to here to 3'. And now you have 3', which goes all the way over to here to 5'. So you have your anti-parallel strands. And what we're seeing here is this double helix of DNA is starting to be unwound. So if you want to duplicate it, if you want to make more copies, you can't do that while it's tightly wound up in a helix. So this DNA molecule has to be unwound. And you can think of it like a piece of rope or a telephone cord that's twisted around itself. You have to really twist it to open it back up into what we call a replication fork. So you're going to open this bubble up in the DNA, and the molecule that's going to do that is an enzyme known as DNA helicase. And if you remember the enzyme terminology that we used, ACE means it's an enzyme, it's DNA helicase. It's an enzyme that's designed to spin DNA. So by spinning that DNA, it's going to unwind and open up this replication fork. The opening of the replication fork is important for all of the processes I'm about to describe. So once you have this fork open, you have a second enzyme which comes in and binds to the DNA molecule. And this enzyme is known as DNA polymerase. So again, ACE, it's an enzyme. DNA polymerase, it makes polymers of DNA. Now DNA polymerase is a very special directed molecule. It can bind DNA and move from the 3' end of the DNA towards the 5' end of the DNA. And as it does this, it's going to be creating new DNA that matches the template 5' to 3'. So you can see, even though this blue strand is going 5' to 3' this way, the DNA polymerase is going in the opposite direction, making a new strand 5' to 3' that's in red. Now, this is called the leading strand for reasons that will become apparent in a second. So if DNA polymerase can move 3' to 5', when it is on the leading strand, it can continue moving, right? As DNA helicase opens this molecule up, the polymerase can continue to move. It can continue to go. It can be processive, making this new molecule. Now on the other side of the replication fork, what we call the lagging strand, this DNA helicase is opening things up, but remember now this blue goes 3' to 5'. So the molecule can't just, the DNA polymerase can't just follow along right into the opening of the fork. The polymerase actually has to go in the opposite direction. And because it goes in the opposite direction, it can only do little bits at a time. And that's all you need to understand for now. It can do little bits at a time. We call these Okazaki fragments after Dr. Okazaki, who discovered these fragments on the lagging strand of DNA. Now, as these little strands are formed, they're patched up later with ligase. But I just want you to appreciate that extra steps are required to get these two strands of DNA to be formed. So the leading strand is the easy one. The polymerase moves right down it. The lagging strand, the polymerase sort of jumps, making the Okazaki fragments, which will be patched later with DNA ligase, which again is an enzyme that ligates, puts together pieces of DNA. So you have the leading strand, you have the lagging strand. Sometimes people call these strands the Watson strand and the Crick strand after James Watson and Francis Crick. Those are just vernacular that you might hear: Watson strand, Crick strand, leading strand, lagging strand. Always remember 5' to 3' is the order by convention that we show a strand if you see nothing else. The last piece of detail I want you to know about replication, really I want you to know that replication occurs by DNA polymerase, but I want you to realize that errors are possible in this process. And this is going to become important when we start talking about evolution. It's possible for DNA polymerase to make a mistake. Now there are a lot of mechanisms, and you learned about some of these that occur in G2 that are able to proofread and fix these mistakes, but mistakes are always possible based on the way this molecule works and based on the amount of DNA that has to be replicated every time a cell divides. So in conclusion, let me just give you a few summary ideas that I want you to take away from this. We talked about the molecule of DNA. We talked about how the molecule of DNA needs to be replicated. When it's replicated, there are specific rules that allow the replication to occur. And those rules are such that adenine always binds to thymine, and guanine always binds to cytosine. A always goes with T, G always goes with C. The other thing that's important for you to realize is that the process of DNA replication is semi-conservative. So one strand is split into two. Each half is used as a template to make the new strands of DNA, and so on and so on and so on every time the DNA is replicated. If you can remember a few of the details of the Michelson-Stahl experiment, that's great. That was a really beautiful demonstration that DNA replication is semi-conservative. But as long as you can remember that DNA is used as a template to make more copies of itself, and that's a good place to be. The most critical enzyme for the DNA replication that occurs is DNA polymerase. So it makes new polymers of DNA. This enzyme will become important later when we talk about bioengineering, because scientists are actually able to use DNA polymerase to make their own molecules of DNA in a laboratory. Other enzymes we talked about are DNA helicase, which unwinds the strand of DNA so that it can be replicated, and DNA ligase, which is used to patch together those Okazaki fragments. So it can put together two strands of DNA that are near each other but not connected. The final thing I want you to realize is that errors are possible in DNA replication. But the most important things to know about DNA replication is that it occurs very quickly. It's a very fast process, and it occurs very accurately. So it's important for the cell that it's able to replicate its DNA very quickly, but also replicate that DNA with a high degree of fidelity. That DNA is accurate every time it makes a copy, because that's what's going to sustain that cell and keep that cell growing in the way that it's supposed to. So DNA replicates with pairing rules, A, T, G, C. The process is semi-conservative. The most critical enzyme is DNA polymerase, and DNA replication has to be fast and has to be accurate for the cell to survive and survive well.